Hi, I'm David and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. Question 1 is about the Asiatic lion. A. Outline the role of the IUCN. IUCN is an organization that makes the Red List. It is a list to categorize organisms according to their conservation status. Organisms are placed into categories such as threatened, endangered, and extinct. This information can be used for educational purposes and for spreading awareness. They also provide a global forum for sharing expertise. Sometimes, IUCN provides advice about conservation issues as well. B talks about the Maori community that coexisted with the Asiatic lions. Suggests why the Maori place old and weak cattle at the edges of their cattle enclosures. They do this so that the lions can easily kill the old and weak cattle for food. This can help to prevent the rest of their cattle from falling prey to the lion so the remaining cattle can survive. C. Describe how IVF can be used with an endangered species such as the Asiatic lion. Firstly, the female is given a hormone. You can name the hormones if you do know about them. This is to stimulate superovulation. Superovulation means the maturation and the release of multiple mature oocytes. Then, the secondary oocytes are harvested. It is done by using a fine needle guided by ultrasound. The oocytes are then incubated with semen. Fertilization may occur. The zygotes are cultured for several days until they reach the embryo or blastocyst stage. The embryo will then be placed in the uterus of the female or surrogate mother. A1. Outline how yeast carries out respiration in anaerobic conditions. When answering this question, please ensure that the sequence of your points is logical. Glycolysis occurs as usual in anaerobic conditions. Glucose is converted to pyruvate. Decarboxylation occurs where carbon dioxide is formed. Pyruvate is converted to ethanol. Then, ethanol is reduced to ethanol. In this step, reduced NAD is oxidized to NAD. You can also say that it leads to the regeneration of NAD. There is a mark for AVP. For example, state the names of the enzymes involved in the process. 2. Explain why respiration in anaerobic conditions is an advantage to yeast. The answer is a general explanation of why anaerobic respiration can benefit an organism. It allows them to survive in the absence of oxygen. This is because ATP is still produced in glycolysis. Glycolysis can continue even though the other stages of aerobic respiration have stopped. The reason is that NAD is recycled when ethanol is reduced to ethanol. This passage details two methods used in the production of bioethanol. They are the high temperature method and the lower temperature method. In B, you have a graph comparing the concentration of starch and ethanol using non-GM and GM yeast. You are asked to describe the trends in the data for ethanol production by the GM strain compared to the non-GM strain. Firstly, make a general statement to show the difference. GM strain has a higher concentration of ethanol, or GM strain has a steeper increase in the concentration of ethanol. Then, mention the similarity. Both increase initially, but then level off. Lastly, use comparative figures to support one of your points. For example, for the first point, you can quote the maximum concentration of ethanol when the different strains are used. C. Suggests why using the lower temperature method has a similar rate of production of ethanol to the higher temperature method. Notice that this question talks about the higher and lower temperature methods mentioned in the passage. It is not about GM and the non-GM yeast in B. The rate of production is similar, as similar or the same concentration of starch is hydrolyzed. So, there will be a similar concentration of glucose available to yeast. The enzymes probably work at the same rate in both methods, leading to a similar production rate. Another possibility is that GM yeast cells produce more enzymes than the ones added in the high temperature method. D. Suggests one reason why the high temperature method is expensive to carry out. Since a higher temperature is needed, more fuel or electricity is required. Another thing is that they need to add enzymes on top of the yeast, so this leads to extra cost. There is a mark for AVP. For example, the whole method takes longer, so maintenance and labor costs would be higher as well. Question 3a shows a diagram of a chloroplast. 1. Identify the structure that contains genes that code for some of the enzymes used in photosynthesis. It is D, as D is the circular DNA. The site of synthesis of some of the enzymes used in photosynthesis. It is B, as B is a ribosome. 2. Explain how grana are adapted for their specific role in photosynthesis. The command word explain means you can't just state the feature. Each of your points must consist of a feature and an explanation of how this feature helps in photosynthesis. Number 1. There are many thylakoids. This is to increase the surface area. You can also explain that the large surface area maximizes the absorption of light energy. The thylakoid membrane contains many photosynthetic pigments to absorb light energy. They also contain electron carriers or ETC to transfer energy from excited electrons. You can find many photosystems on the membrane. They act as the light harvesting structures. Thylakoid space or lumen can form a proton gradient. Next, the thylakoid membrane is impermeable to protons. This is to maintain the proton gradient for chemiosmosis. On the membrane, there is ATP synthase to make ATP. Lastly, they contain oxygen evolving complex or OEC for the photolysis of water. B is about an experiment on the effect of changing light conditions on the pH of the chloroplast stroma. Figure 3.2 shows how the pH changes in the stroma in dark and light conditions. 1. Describe the results shown in Figure 3.2. Firstly, give the general statement. The pH is higher in the light than in the dark. Then, give the details of each part, how it increases, levels off, and decreases. The graph also shows that the pH fluctuates throughout the experiment. The last mark is given to any comparative figures to support the point. For example, to support marking point number 1, you can quote in the pH range in the dark and light conditions. For this point, remember to place them right after the point they are supporting. Do not place the data quote in the end. 2. Discuss how the results in Figure 3.2 support that chemiosmosis occurs during photophosphorylation. To answer this question, you must know that chemiosmosis occurs due to the proton gradient, and protons can decrease the pH of the solution. The stroma has a higher pH when protons move out of the stroma into thylakoid space. This leads to increased proton concentration in the thylakoid space. Then, proton diffuses back into the stroma through ATP synthase, leading to the production of ATP. There is a mark for AVP. You can say that the results does not support the theory as pH changes may due to other factors. The graph cannot prove that it is definitely due to the movement of protons. In question 4, we have a table comparing the two forms of hemophilia, A and B. A. Genetic engineering is used to make recombinant human proteins to treat people with hemophilia A and hemophilia B. Outline the principles of genetic engineering. Outline means that you only need to provide the main steps in your answers. You don't have to give the details of each step. Also, pay attention to the sequence of your points. The arrangement should be logical. You can get a mark by giving the goal of the technique, which is to manipulate DNA to modify an organism's characteristics. Gene, allele or DNA is obtained using restriction enzyme. You can also use reverse transcriptase to make cDNA using mRNA. The gene is inserted into a vector or plasmid. Ligase is used for this step. The resulting recombinant plasmid is inserted into the host cell or bacterium. Then, clone or multiply the host cell in a fermenter. When the gene is expressed, factor IX is made. There is a mark for AVP. For example, artificial or chemical synthesis of a new gene to be inserted into the plasmid can be done. B talks about the possibility of gene therapy using a virus. B1. Assess which form of hemophilia they should try to treat first, and whether they should attempt to treat it with gene therapy. Explain your reasoning. Both sides of the argument are accepted in this question if you correctly reason your opinion using the information provided. Let's discuss which form of hemophilia they should treat first. If your answer is hemophilia B, you can use the gene length to support your choice. The F9 gene is only 1.6 kilobase pair. It is small enough to fit into the virus with a genome of 4.7 kilobase pair. If your choice is hemophilia A, it is probably because it is more common than B. Then, take your stand on the attempt to do it. If you think they should not attempt gene therapy, you can say that recombinant proteins can manage the disease, so gene therapy is not necessary. Gene therapy using viruses may have a lot of risks, such as side effects and allergic reactions. If you think that it should, you can reason that it provides a cure. The other treatments, such as infusion of clotting factors regularly, can harm the patients as well. 2. Explain why a promoter has to be introduced as well as the desired gene. A promoter is the sequence for binding RNA polymerase or transcription factors. The binding switches the gene on, so the gene is expressed. You can also say that the promoter allowed the transcription of the gene. The promoter can control the expression of the gene so it occurs at all the time or at the right time, in sufficient quantities, and in the correct tissue. 3. Some individuals taking part in gene therapy trials have been naturally exposed to the virus carrying the functioning gene so that their blood already contains antibodies to the virus. Predict how this will affect the success of the gene therapy treatment. Exposure to the virus causes an immune response and the production of memory cells. So, when the same virus is used in gene therapy, they can produce antibodies rapidly by secondary immune response. This may destroy the virus or the viral vector. So, it limits the delivery of the gene into the cells. The person's immune system may also destroy the GM cells or the cells that have successfully taken up the DNA. C. State 2 possible advantages of using gene editing as a method of treating hemophilia. Firstly, gene editing is precise or accurate, using guide RNA to find the targeted gene to be cut by Cas9. A patient's own gene can be edited or corrected, so no introduction of gene or allele is needed. Promoter also does not need to be introduced. Gene editing does not involve viruses, so there is less or no risk of cancer or immune response. Question 5 talks about a plant species that grow at high altitude at the tops of mountains in New Zealand. A gives us the detail of PCR. You need to describe what happened to the DNA at each temperature. The first thing we must do in PCR is to break the hydrogen bonds between the complementary strands in a double-stranded DNA. This happens at 95 degrees Celsius. Then, we lower the temperature to 50 degrees Celsius, which allows the annealing of primers to the single-stranded DNA. Then, the temperature is increased to 72, the optimum temperature for tag polymerase. A complementary DNA strand is made as DNA replication occurs. B shows the map of New Zealand. Scientists compare the DNA sequence data for 3 myosotis species. The results are shown here as 3 bullet points. You need to discuss reasons for the results in the 3 species. The first bullet point shows us that M. pygmae populations are geographically isolated by sea between the South Island and the Stewart Island. The second bullet point shows us a similar situation of M. pulvinaris populations as they are found on different mountains. So, the explanation for the genetic differences between the populations of these 2 species are the same. Geographical isolation exists between the populations of both the species. The geographical isolation causes no or little gene flow between the populations. They experience different selection pressures due to the different environments they live in. There might be different or random mutations occurring in each of the populations. The changes in allele frequencies would not be the same for them, leading to different gene pools. So, allopathic speciation could be occurring. The third bullet point shows that there is no geographical isolation in the populations of M. porciana. They do interbreed. Gene flow can occur within or between the populations. These lowland species live in different areas of the same island, so they experience similar selection pressures, which is why they generally show genetic similarities. C. State 2 factors other than natural selection that could give genetic changes in populations of Myosotis. Firstly, the random change of allele frequencies by genetic drift, bottleneck effect, or founder effect. Migration may introduce new alleles into a population. Mutation can cause the arising of new alleles from the existing ones. Artificial selection done by human is a factor as well. Since the command word in the question is state, no elaboration is needed for the points. Question 6 shows the tryptophan operon, another operon found in prokaryotes, beside the lag operon. The diagram shows that transcription of the structural genes does not occur when tryptophan is present. 1. Describe the differences in structure and control between the lag operon and the tryptophan operon. Let's talk about the structure. The TRP operon regulatory genes codes for an inactive repressor, while the lag operon regulatory gene codes for an active repressor. The attenuator in the diagram was not found on the lag operon. The lag operon has 3 structural genes, but TRP operon has 5. Regarding control, the lag operon uses an inducer called allolactose, and the TRP operon uses a repressor, tryptophan. In the lag operon, allolactose binds to and inactivates the repressor, so the repressor cannot bind to the operator. The binding of tryptophan to the repressor has the opposite effect. Lastly, allolactose causes gene to be transcribed or switched on or expressed, and tryptophan causes the opposite. 2. Suggest why structural genes in operons are transcribed together. It is because they share one promoter. The enzymes or proteins or products coded by these genes work together in the same process, so they must be produced together. 3. Describe the differences between the functions of structural genes and regulatory genes. All structural genes code for non-regulatory proteins such as enzyme, structural proteins, rRNA, and tRNA. Regulatory genes code for proteins that control another gene's expression or transcription. They code for transcription factors and repressor proteins. 4 tells you that TRPA, one of the structural genes in the tryptophan operon, codes for the enzyme tryptophan synthase. It catalyzes the formation of the amino acid tryptophan. Explain why tryptophan synthase is an example of a repressible enzyme. This is an example of end product inhibition or negative feedback inhibition. The product of the structural gene binds to and activates the repressor. It means that when tryptophan is produced, it stops or reduces the gene expression, which codes for the enzyme that made it. So, tryptophan synthase is no longer being synthesized. B is about the control of gene expression in eukaryotes. 1. Describe how gibberellin activates gene in plant cells. When gibberellin is produced, it binds to the receptor on the cell surface membrane. This causes the synthesis of enzymes, which leads to DELA breakdown. It is wrong if you say that gibberellin breaks down DELA proteins. DELA no longer binds to transcription factors, so the inhibition is removed. The transcription factor, called PIF, binds to the promoter. Genes involved in growth will be switched on or expressed or transcribed. 2 provides some information on 2 proteins. When JSZ proteins are broken down, MYC proteins are free to bind to DNA and allow transcription to begin. State the term that is used to describe proteins such as MYC proteins. Since MYC allows transcription to begin when it binds to a DNA, it is a transcription factor. Question 7 is about the sex chromosomes found in turkeys. Male turkeys are ZZ and females are ZW. The gene for feather color is located only on the Z chromosome. The dominant allele codes for bronze and the recessive allele codes for brown. A. Define the terms dominant and recessive. Dominant is the allele that always affects phenotype. It is expressed in homozygotes and heterozygotes. In other words, it is always expressed. The recessive allele only affects phenotypes if the dominant allele is absent. It can only be expressed in a homozygote. B wants you to construct a genetic diagram to show the results of a cross between a heterozygous bronze male turkey and a brown female turkey. When you choose the symbol, remember that this is a sex-linked condition, so you must use Z and W in your symbol. Since the feather color gene is only found on the Z chromosome, the symbols will look like this, with the allele as superscript. The male turkey is heterozygous, so its genotype is Z big B and Z small b. The female is brown, so it has a Z small b and a W, which does not contain the allele. Then, separate these alleles into the gametes. Write down all possible combinations when the gametes fuse and match the genotypes with the phenotypes they show. C. Explain how you would carry out a test cross to determine the genotype of a bronze male turkey. In a test cross, we always cross the individual we want to find out with a homozygous recessive individual. Since female turkeys would only have one Z chromosome, we will use a recessive female for the test. Then, by using the outcome of the cross, we can tell the genotype of the bronze male turkey. In this kind of question, you must always state the outcome and describe what do they indicate. Here is a diagram of the proximal convoluted tubule and part of the adjacent blood capillary. A wants you to label where these proteins can be found. The first one is the co-transporter protein. This is the protein that allows glucose to move into the PCT cell together with sodium ions. So, you will find it on the microvilli facing the lumen of PCT. The second one is sodium-potassium pumps. It actively transports sodium from the PCT cell to the tissue fluid, so you will find it on the basal membrane adjacent to the blood capillary. Note that both labels must touch a membrane, not a space. B. Explain how the cells of the proximal convoluted tubule are adapted to carry out selective reabsorption. The word explain means that you must provide the feature and elaborate on how these features or structures help in the process. There are microvilli for large surface areas. It increases the rate of reabsorption. This also means many co-transporters or carrier proteins can be found on the cell surface membrane. Co-transporter proteins can be used as an answer itself. Their presence allowed the secondary active transport of glucose and amino acids into the cell. There are tight junctions between cells, so substances must pass through the cell as there will be no leakage. Many mitochondria are found in the cell. They produce ATP. ATP is used to transport sodium ions into the blood actively. Lastly, the folded basal membrane embeds many sodium-potassium pumps, increasing the sodium transportation rate. In C, a table shows the percentage composition of plasma, glomerular filtrate, and urine. You need to calculate how much the percentage composition of urea has increased in the urine compared with the glomerular filtrate. Since the values are already percentages, you must divide them to know how many times they increase. You don't have to multiply it with 100 again. The first diagram in 9a shows the voltage-gated channels of sodium and potassium ions in the membrane of an exon. The second diagram shows the changes in membrane potential over time. You are asked to complete table 9.1 to match the phases with the states. A is when we have the resting potential. None of those voltage-gated ion channels open, so it is 2. C is when depolarization occurs, hence an increase of membrane potential. The sodium ion channel opens at this time, so it is 1. E is repolarization and F is hyperpolarization. Both occur due to the movement of potassium ions out of the exon, So it is 3 when potassium ion channels open. G is when the resting potential is restored. So we go back to 2, just like in A. B. Describe and explain the role of myelin sheath in the transmission of an action potential. First, Schwann cells wrap around the axon and form the myelin sheath. It insulates the axon and prevents the movement of ions. Due to this insulation, depolarization or action potentials can only occur at nodes of Ranvier. This creates a longer local circuit as the nodes are 1 to 3 mm apart. Action potentials move by saltatory conduction. You can also say that it jumps from node to node. The transmission speed is faster than an unmyelinated axon, which is at 100 meters per second. 10 shows the relationship between blood glucose concentration and blood glucagon concentration in a healthy person. The question wants you to explain the relationship shown in the figure. The first part of the graph shows that the decreasing blood glucose concentration causes an increase in glucagon concentration. In other words, glucagon secretion is stimulated by the low blood glucose concentration. Glucagon will then act on liver cells. The rate of glycogenolysis in the liver cells increases, where glycogen is broken down in liver cells and glucose is formed. It also stimulates gluconeogenesis, where pyruvate is converted to glucose. Glucose is then released into the blood. Blood glucose concentration increases afterwards. Negative feedback occurs when the blood glucose concentration is restored to the set point. In B, we need to complete a passage using the most appropriate scientific terms. Glucagon binds to the receptors because its shape is complementary. This is true for all kinds of ligands and their receptors. Same, matching, or similar are not acceptable to describe this relationship. This binding activates G protein, leading to the stimulation of the enzyme adenylyl cyclase. These steps are needed for the synthesis of the second messenger. That's all for today. If you think my videos are useful, don't forget to like, comment and subscribe. Thank you for watching and see you again soon.